a boundary so sharp that geologists can trace it in rock formations across 6 continents. Below that line, the bones of creatures that ruled the land for 160 million years. Above it, silence and the slow tentative beginnings of a different world. We call it the End Cretaceous extinction event. 66 million years ago, something struck the Earth with a force that the human mind can barely register as real. The object was roughly the size of a small city 10 to 15 kilometers across. It was traveling at approximately 70,000 kilometers per hour when it made contact with shallow tropical water near what is now Mexico's Yucatán Peninsula. The energy released in the first moments of impact was equivalent to 1 billion nuclear warheads detonating simultaneously. The ground itself became wave, rock turned briefly into fluid. The impact crater that formed today, buried beneath limestone and sediment, is roughly 180 kilometers wide. It still holds the shape of that moment, but the impact itself was only the beginning. In the weeks and months that followed, the world entered a condition that some researchers have compared to a nuclear winter. Enormous quantities of sulfur and carbon dioxide were blasted into the upper atmosphere. Soot from continent-wide wildfires joined the cloud. Sunlight weakened. Photosynthesis collapsed. The base of the food chain disintegrated, and the collapse rippled upward through every ecosystem on Earth. Approximately 3/4 of all species on the planet were gone within a geological instant. The non-avian dinosaurs vanished. The mosasaurs, the pterosaurs, countless species of marine invertebrates, forms of life that had existed for tens and hundreds of millions of years, that had survived previous catastrophes, that had spread to every continent and every ocean gone, within a span of time that is almost nothing on the geological scale. And yet life continued. Small, warm-blooded creatures that had lived in the shadow of giants now found themselves inheriting an open world. Mammals diversified explosively, flowering plants recovered, then flourished. The birds, descendants of a lineage of small feathered theropods, expanded into ecological roles that had suddenly become vacant. The world that emerged from that catastrophe eventually gave rise to us, but consider what the planet had been before. Consider the almost incomprehensible depth of time that preceded that moment of impact. 4.5 billion years of geological history, oceans forming and evaporating and forming again, continents colliding and breaking apart. Life emerging from chemistry and then complexifying over hundreds of millions of years until the world was filled with organisms of stunning variety. To understand the Earth we live on, we have to be willing to travel backward through time, far backward, to conditions so alien that our planet would be unrecognizable. This is the story of how the Earth became what it is. How life found its footing on a hostile, volcanic, radiation-drenched world. How simple organisms quietly transformed the atmosphere of an entire planet. How catastrophe and recovery have been the recurring rhythm of our world's long history. This is the chronicle of prehistoric Earth, begin at the very beginning. 5 billion years ago, there was no solar system in its place, a vast cloud of gas and dust. A nebula drifted through a region of the Milky Way galaxy. The material in this cloud was the legacy of previous generations of stars. Stars that had burned through their fuel, collapsed, exploded, and scattered the products of their nuclear furnaces across interstellar space: carbon, oxygen, iron, silicon. The raw materials of planets and of life had already been forged long before the sun existed. At some point, something disturbed this cloud. Perhaps the shockwave from a nearby supernova. Whatever the cause, gravity began to win. The cloud contracted. As it contracted, it spun. As it spun, it flattened into a disk. At the center, material gathered and compressed until pressure and temperature became extreme enough to ignite nuclear fusion. A new star was born. Around that star, within the rotating disk of remaining material, smaller concentrations of matter were forming. Dust grains collided and stuck together. Pebbles accumulated into rocks. Rocks accumulated into boulders. Over millions of years, through the process we call accretion, these fragments grew into planetesimals, bodies kilometers across, their surfaces scarred by constant impacts. And the planetesimals grew too, merging in collisions of staggering violence, until a handful of large bodies dominated the inner solar system. 4,540,000,000 years ago, one of those bodies had grown large enough to hold its own gravity, to pull in material from its surroundings, and to begin the long process of becoming a world. That world was Earth. In its earliest incarnation, the Earth we live on would have been utterly unrecognizable. The surface was largely molten. Heat from the continuous rain of impacting bodies, combined with the decay of radioactive elements in the interior, kept the rock in a state of perpetual flux. There was no solid crust to speak of, only a slowly cooling magma ocean, its surface hardening into thin plates that were constantly broken and remelted. The sky above was not blue. It was orange and yellow and dense with volcanic emissions, carbon dioxide, water vapor, hydrogen sulfide, sulfur dioxide, methane, ammonia. The kind of atmosphere that would kill a human being in minutes. This period, the Hadean Aeon, named for the ancient Greek conception of the underworld, lasted from the formation of the Earth until roughly 4 billion years ago. The name is appropriate. The young Earth was, by any modern standard, a kind of hell. Surface temperatures were extreme. Ultraviolet radiation from the young sun was intense, with no ozone layer to block it. Impacts from leftover solar system material were frequent and sometimes catastrophic. And yet even in this environment, processes were underway that would eventually make life possible. The heavier elements, iron, nickel, sank toward the center of the young Earth, forming the core. Lighter silicate materials rose toward the surface, beginning to form what would eventually become the continental crust. As the planet cooled, water vapor in the thick atmosphere began to condense. It rained. It rained continuously for perhaps millions of years. The rainwater gathered in the low-lying regions of the cooling surface, and the first oceans, hot, acidic, and entirely lifeless, began to form. One of the most fundamental questions in the history of science is, how old is the Earth? For most of human history, the answer seemed obvious and was wildly, spectacularly wrong. In medieval European thought, the age of the world was calculated from genealogies and calendars in religious texts. Scholars arrived at figures of a few thousand years. As late as the 17th century, careful biblical chronologists were producing estimates in the range of 6,000 to 10,000 years. These numbers felt precise and well-grounded because no one yet had a framework for thinking differently. The first systematic challenges came from geology. As naturalists began to study rock formations in the 18th and 19th centuries, they encountered a problem. The processes that form sedimentary rock, the slow deposition of particles in water, layer by layer, proceed at measurable rates. When geologists looked at the thickness of ancient sedimentary sequences and tried to work backward, the implied timescales were enormous, hundreds of millions of years. Far more than any theological chronology allowed. The physicist William Thomson, later given the title Lord Kelvin, approached the problem from a different direction. In the 1860s and 1870s, he calculated how long it would have taken a molten Earth to cool to its current temperature. Using the best physics available to him, he arrived at an estimate of somewhere between 20 and 40 million years. This seemed like a reasonable reconciliation between the geological evidence and the constraint that the Earth couldn't be older than the Sun could sustain. But it still felt too short for many geologists, who could see in the rock record evidence that seemed to demand far longer timescales. The resolution came unexpectedly from a discovery that Kelvin could not have anticipated. In 1896, Henri Becquerel discovered radioactivity. Within a few years, Ernest Rutherford and Frederick Soddy worked out that radioactive elements decay at fixed measurable rates that are entirely unaffected by temperature, pressure, or chemistry. This was, in hindsight, fatal to Kelvin's calculation. If the Earth's interior contains radioactive elements generating heat, then the planet hasn't simply been cooling down from an initial molten state, it has had an ongoing internal heat source throughout its history. Kelvin's model was built on a false assumption. But radioactivity gave scientists something far more valuable than a correction to Kelvin's calculation. It gave them a clock. Radioactive decay is so predictable, so immune to external conditions, that it can be used to date ancient rocks with remarkable precision. By measuring the ratio of parent isotopes to daughter isotopes in a rock sample, scientists can calculate how long the decay has been occurring, and therefore how long ago the rock formed. When this technique was applied to the oldest terrestrial rocks, and then to meteorites, fragments of the early solar system that have been orbiting the Sun largely unchanged since its formation, a consistent age emerged: 4.5 billion years. The Earth is 4,540,000,000 years old, a number that only became possible to grasp once humanity had developed the tools to measure it. Now let us return to the early Earth and to one of the most consequential events in our planet's history. At some point during the Hadean Eon, the timing is still debated, but probably in the first few 100 million years of Earth's existence, the planet experienced a collision of extraordinary violence. Another planetary body, roughly the size of Mars, struck the proto-Earth at an oblique angle. The impactor, which planetary scientists have given the name Theia, was partially absorbed into the Earth. But a massive quantity of vaporized and molten material was ejected into orbit. This debris cloud stretched around the Earth in a glowing ring, cooled and aggregated over a relatively short period, perhaps just a few thousand years, into a single body. The newly formed Moon was much closer to Earth than it is today. Some models suggest it may have been as close as 20,000 kilometers, compared to the nearly 400,000 kilometers of its current distance. At that proximity, the Moon would have appeared enormous in the sky, filling a much larger portion of the visual field than the Sun. The tidal forces it exerted on the early Earth were enormous. Ocean tides would have been dramatically more powerful, potentially reaching heights of hundreds of meters in some locations. The Earth's rotation was faster, giving the early planet a day of perhaps only 6 hours. Over billions of years, tidal interactions have gradually transferred angular momentum from the Earth's rotation to the Moon's orbit, causing our satellite to slowly spiral outward. This process continues today. The Moon recedes from Earth at a rate of about 3.5 centimeters per year, faster than a fingernail grows, but geologically significant over deep time. The Moon's presence has had consequences far beyond aesthetics. The gravitational relationship between Earth and Moon stabilizes the axial tilt of our planet, the angle at which Earth's axis is inclined relative to its orbit around the Sun. This tilt, currently about 23.5 degrees, is what gives Earth its seasons. Without the Moon's stabilizing influence, models suggest that Earth's axial tilt might vary chaotically over millions of years, producing climatic swings of extraordinary severity. The relatively stable seasons we experience, season after season and millennium after millennium, owe something to a collision that happened 4.5 billion years ago. There are alternative hypotheses for the Moon's origin. One suggests that the Moon formed simultaneously with Earth from the same disk of material rather than from a later impact. Another proposes that the Moon formed elsewhere in the solar system as an independent body and was later captured by Earth's gravity. A third model considers the possibility that the early Earth rotated so rapidly that material was spun off from the equator and coalesced into the Moon. Each of these scenarios has difficulties and evidence that both supports and challenges it. The giant impact hypothesis remains the best supported. But the details of exactly how the Moon formed continue to be refined as scientists study lunar samples and run increasingly sophisticated computer simulations. What we know with confidence is that the Moon is there, that it has been there for most of Earth's history, and that its influence on our planet has been profound. As the Hedin gave way to the Archean Eon roughly 4 billion years ago, Earth was still a dramatically different world from the one we know, but significant changes were underway. The surface had cooled enough for a permanent solid crust to form, though it was thinner and more geologically active than modern crust. The atmosphere was still oxygen-free, dominated by nitrogen, carbon dioxide, and methane, with some water vapor. There was no ozone layer. The young sun, paradoxically, was only about 70% as luminous as it is today, but the thick greenhouse atmosphere of the early Earth compensated for this, keeping surface temperatures above freezing. And somewhere, in conditions that we still do not fully understand, chemistry crossed a threshold. Life began. The oldest direct evidence we have of life on Earth dates to roughly 3.5 billion years ago, stromatolites found in the Pilbara region of Western Australia. Stromatolites are layered structures built by communities of microorganisms, primarily cyanobacteria, which trap and bind sediment particles as they grow. In cross-section, ancient stromatolites show the characteristic laminated pattern of successive microbial mats accumulated over vast spans of time. They look from the outside like rounded or columnar rocks. They are among the most extraordinary objects on Earth, humble in appearance, profound in their implications. Living stromatolites still exist today, in a handful of highly saline environments where the salt concentration is too high for the organisms that would otherwise graze on them. The shallow, clear waters of Shark Bay in Western Australia host perhaps the world's most famous living stromatolite communities. Looking at them is as close as we can come to seeing the ancient biosphere with our own eyes. But the organisms that built those early stromatolites were doing something that would, over hundreds of millions of years, transform the entire planet. They were photosynthesizing. The cyanobacteria, or their ancestors, were capturing sunlight and using its energy to split water molecules and fix carbon dioxide, producing organic carbon for their bodies and releasing oxygen as a byproduct. For most of the Archean, this oxygen was immediately consumed, absorbed by iron dissolved in the ancient oceans, reacting to form iron oxides that settled to the seafloor. These ancient iron formations are now mined worldwide as iron ore. They are, in a very real sense, the chemical record of early photosynthesis. For hundreds of millions of years, the oxygen produced by early life was captured almost as fast as it was generated, but gradually the iron in the oceans was depleted, and when the chemical sink was gone, oxygen began to accumulate in the atmosphere. This transition, which occurred roughly 2.4 billion years ago during what geologists call the Great Oxidation Event, was one of the most dramatic environmental shifts in Earth's history. For organisms that had evolved in an oxygen-free world, oxygen was a poison. The accumulation of this reactive gas in the atmosphere was, in a very real sense, the first mass extinction, a catastrophe for the anaerobic life forms that had dominated Earth's early biosphere. But the consequences went further. Methane, which had been a major component of the early atmosphere, reacts with oxygen. As oxygen levels rose, atmospheric methane was rapidly destroyed. Methane is a powerful greenhouse gas, and its removal triggered a dramatic cooling event. The Earth entered what is known as the Huronian glaciation, a period of extreme cold that lasted for roughly 300 million years. beginning around 2.4 billion years ago and ending perhaps 2.1 billion years ago. Evidence suggests that ice may have extended to very low latitudes during this period, possibly reaching all the way to the equator in what some researchers have called a snowball Earth event. The planet that emerged from the Huronian glaciation was, in some fundamental ways, a new Earth. The atmosphere contained free oxygen. Aerobic metabolism, the ability to use oxygen to extract energy from organic molecules, had become advantageous, and over the following hundreds of millions of years, new forms of cellular complexity would emerge that would eventually make multicellular life possible. It is worth pausing here to appreciate the sheer timescale involved. From the formation of Earth to the Great Oxidation Event is roughly 2 billion years. From the Great Oxidation Event to the emergence of the first animals is another billion years or more. The entire history of complex multicellular life on Earth, the era of visible macroscopic organisms, from the first animal-like forms to the present day, occupies only about 600 million years. That is a long time by any human measure. But it represents only the final 12% of our planet's history. For nearly 90% of Earth's existence, life consisted entirely of single-celled organisms, bacteria and archaea, simple in structure but extraordinary in their chemical versatility, colonizing environments from hot springs to deep ocean vents to the interior of rocks, were the dominant expressions of life on Earth for most of its history. And then, with a kind of geological suddenness, everything changed. 540 million years ago marks the beginning of the Cambrian period, and with it, the Phanerozoic Eon, the eon of visible life. Within a geologically short span of tens of millions of years, the fossil record shows an extraordinary diversification of animal life. This event, the Cambrian explosion, produced representatives of most of the major animal body plans that exist today. Eyes, hard shells, articulated limbs, predation. The relationships between predator and prey, between organisms competing for space and resources, had apparently driven an evolutionary arms race that generated tremendous biological diversity in a remarkably short time. The ocean of 500 million years ago would look profoundly strange to modern eyes. Trilobites, now extinct, but once among the most successful of all animal groups, crawled across the seafloor and swam in the water column. Anomalocaridids, filter-feeding giants that could reach a meter or more in length, cruised the open water. Soft-bodied worms and mollusks populated the seafloor. Reefs built not by coral but by sponges and other organisms arose from the shallow seafloor. The land, by contrast, was still mostly bare. The continents of the early Paleozoic were not arranged as they are today. Much of the landmass of the southern hemisphere was gathered into a single supercontinent, Gondwana, which included the land that would eventually become Africa, South America, Antarctica, Australia, and the Indian subcontinent. North of it, smaller continents occupied tropical and equatorial regions. The combination of these configurations, together with the absence of land plants, meant that terrestrial erosion patterns were radically different from today. There were no forests to hold soil in place, no root systems to break rock. Rivers ran through landscapes of bare rock and sediment. The Ordovician period, from about 485 to 443 million years ago, saw continued diversification of marine life. Then, at the end of the Ordovician, the second largest mass extinction in Earth's history struck. Glaciation, possibly linked to the movement of Gondwana over the South Pole, caused sea levels to drop dramatically. The shallow marine environments where so much life was concentrated shrank. Estimates suggest that perhaps 85% of marine species were lost. Life recovered. It always has. The Silurian period, which followed, saw the colonization of land accelerating. The earliest land plants, descendants of green algae, had begun to appear in the late Ordovician, but it was during the Silurian that they began to diversify significantly. Simple, rootless, leafless plants slowly spread across coastal and riparian zones. At the same time, some arthropods, relatives of today's centipedes and scorpions, were making the transition to terrestrial life. The arthropods were well suited for this. Their external skeletons helped prevent desiccation, and their book lungs or tracheal systems allowed them to breathe air. The Devonian period, from about 418 to 359 million years ago, is sometimes called the Age of Fishes. The oceans and freshwater systems of the Devonian were dominated by fish of extraordinary variety, armored plecoderms, early sharks, lobe-finned fish, whose descendants would eventually give rise to all land vertebrates. The largest Devonian predator known, Dunkleosteus terrelli, was an armored fish that may have reached 9 meters in length, with a bite force that some estimates place among the most powerful of any animal in Earth's history. On land, the Devonians saw the appearance of the first forests. Trees, actual trees, with woody trunks and leaves, began to spread across the continents for the first time. The earliest large trees, such as Archaeopteris, created shaded forest environments that would have been recognizable in some ways to a modern eye, even if nothing familiar walked beneath them. The colonization of the land by vascular plants had a profound effect on atmospheric carbon dioxide levels, drawing down CO2 through weathering reactions and organic carbon burial, and contributing to a global cooling trend. At the end of the Devonian, another major extinction event struck, actually a series of extinction pulses spread over several million years. Marine life was particularly hard hit, especially the reef-building organisms of the time. Then came the Carboniferous period, stretching from roughly 359 to 299 million years ago. The name comes from the vast deposits of coal that were laid down during this time, and understanding why those coal deposits formed tells us something remarkable about the biology and ecology of the Carboniferous world. The forests of the Carboniferous were dominated by tree-sized lycopsids, relatives of today's club mosses, but reaching heights of 30 meters or more, and giant horsetails and seed ferns. These forests spread across the tropical lowlands of the Carboniferous world, which at that time straddled the equator. The climate was warm and humid. The forests were lush beyond anything in the modern world. But here is the key: when those trees died, their wood did not rot. Or rather, it rotted very slowly and incompletely. The organisms capable of efficiently decomposing lignin, the tough structural polymer that gives wood its strength, had not yet evolved or were not yet widespread. So the fallen trees accumulated layer after layer of organic material piled up in vast swamps. Compressed and buried over millions of years, this organic material eventually became coal. The fossil fuels we have burned since the Industrial Revolution are, in a very real sense, the preserved sunlight of Carboniferous forests, energy captured from the sun 300 million years ago, locked into carbon and released again by combustion in our power plants and engines. The atmosphere of the Carboniferous was rich in oxygen, perhaps reaching 35% by volume at its peak, compared to 21% today. This elevated oxygen level had dramatic consequences for the organisms that lived then. Insects, which breathe through a system of passive tubes, can grow larger when atmospheric oxygen is higher, because oxygen penetrates deeper into their bodies. The Carboniferous produced insect giants unlike anything alive today. Meganeuropsis permiana, a dragonfly-like insect from the late Carboniferous and early Permian, had a wingspan of nearly 70 centimeters, larger than some modern hogs. Arthropleura, a millipede-like arthropod, reached lengths of 2.5 meters. The Carboniferous swamps were populated by creatures that would seem monstrous by modern standards. Reptiles appeared for the first time during the Carboniferous. having evolved from amphibian ancestors that had themselves evolved from lobe-finned fish during the Devonian. The critical innovation of the reptiles was the amniotic egg, an egg with a protective membrane and shell that could be laid on dry land, freeing vertebrate reproduction from its dependence on water. This was a liberation of enormous significance, allowing reptiles to colonize environments far from lakes and rivers. The Permian period followed the Carboniferous, lasting from roughly 299 to 252 million years ago. The climate became drier and more seasonal as the continents, still assembling into the supercontinent Pangaea, created vast continental interiors far from oceanic moisture sources. The coal swamp forests largely disappeared. Deserts spread across the interior of Pangaea. Among the fauna, the therapsids, a group of reptile-like animals that included the direct ancestors of mammals, became increasingly dominant. Some therapsids had evolved warm-bloodedness, complex social behaviors, and dentition patterns that foreshadowed the teeth of modern mammals. And then came the end of the Permian, the greatest mass extinction in the history of complex life. Approximately 252 million years ago, something happened that pushed life to the very edge of extinction. The precise trigger is still debated. Massive volcanism in what is now Siberia. The eruption of the Siberian traps released enormous quantities of carbon dioxide and sulfur dioxide over a span of perhaps 1 million years. Global temperatures rose dramatically. Ocean chemistry was disrupted. The oceans may have become significantly depleted in oxygen. Whatever the cascading sequence of events, the result was devastating. Approximately 96% of all marine species and 70% of terrestrial vertebrate species went extinct. Entire groups that had been dominant for hundreds of millions of years simply vanished. The recovery from the end Permian extinction was slow and uneven. For several million years into the Triassic period, life remained relatively sparse and ecologically simplified. But life did recover, and as it recovered, it diversified along new lines. The Mesozoic era, the Triassic, Jurassic, and Cretaceous periods together, spanning from roughly 252 to 66 million years ago, is dominated in popular imagination by the dinosaurs. And rightly so, the dinosaurs were an extraordinary evolutionary success story, diversifying to fill ecological roles from tiny insectivores to the largest land animals that have ever lived. But the Mesozoic was not just about dinosaurs. It was also the era when the continents began to break apart from Pangaea into configurations that by the end of the Cretaceous were beginning to resemble the modern world. It was when the first flowering plants appeared and rapidly came to dominate terrestrial vegetation. It was when the first true birds evolved from small feathered theropod dinosaurs, and when the first mammals, small, largely nocturnal creatures living in the shadow of the dinosaurs, were diversifying quietly and patiently waiting for their moment. The Jurassic period in particular produced the iconic large sauropod dinosaurs, massive long-necked herbivores like Brachiosaurus and Diplodocus that represent the pinnacle of terrestrial gigantism. Diplodocus reached lengths of perhaps 27 meters. Argentinosaurus, from the Cretaceous of South America, may have been even larger, estimates of its length range up to 35 meters, with mass estimates exceeding 70 metric tons. How these animals supported their own weight, how they breathed, how they regulated their temperature and circulation, these questions continue to be refined as paleontologists develop new analytical tools and find new specimens. The Cretaceous period brought further changes. The supercontinent continued to fragment. North America was split by the Western Interior Seaway, a shallow eperic sea stretching from the Gulf of Mexico to the Arctic. The world was warmer than today, with no polar ice caps. Atmospheric carbon dioxide was higher, sea levels were higher. The continents were arranged differently enough. that the fauna and flora of the Cretaceous world were, in many respects, organized along geographic lines that no longer exist. It was in the isolation of different continental masses that some of the most intriguing evolutionary stories of the Cretaceous unfolded. The eastern portion of North America during the late Cretaceous, the landmass that paleontologists call Appalachia, was separated from the western portion by the interior seaway. For millions of years, the animals of Appalachia evolved in relative isolation, following evolutionary trajectories distinct from those of the better-known western landmass called Laramidia. Laramidia, with its richer fossil record, has given us the iconic large dinosaurs of the classic Cretaceous, Tyrannosaurus, Triceratops, Edmontosaurus. But Appalachia has remained largely mysterious because the geological conditions there were less favorable to fossil preservation. Recent decades have begun to fill in some of those gaps. Researchers working in formations from New Jersey to Alabama have recovered fragmentary but significant specimens suggesting that Appalachia hosted its own distinctive fauna, its own tyrannosaurs, its own hadrosaurs, its own horned dinosaurs. Analysis of some of these specimens has revealed that the tyrannosaurs of Appalachia were anatomically distinct from their western relatives in ways that reflect millions of years of separate evolution. Different proportions of limbs, different skull architecture, characteristics that suggest a related but genuinely divergent lineage. One such tyrannosaur, informally nicknamed by researchers who recovered it, inhabited the shallow coastal environments of late Cretaceous Appalachia roughly 85 million years ago, nearly 20 million years before the extinction of the non-avian dinosaurs. Its remains are fragmentary, as is often the case with Appalachian fossils. But they are sufficient to establish that it was a large predator similar to but distinct from the western tyrannosaurs that appear in textbooks and museum halls. The existence of such animals reminds us how incomplete our picture of Mesozoic life remains, and how much may yet be recovered from formations that have been understudied. Among the questions that paleontologists have debated for decades is the social behavior of tyrannosaurs. The popular image of Tyrannosaurus rex as a solitary predator is deeply embedded in the cultural imagination, but the evidence is more complicated. In several locations across western North America, multiple tyrannosaur individuals have been found in the same geological horizon, buried together under conditions that suggest a shared death event. A site in southern Alberta has yielded dozens of Albertosaurus specimens, all apparently killed by the same flooding event. Sites in Montana and Utah have produced similar multi-individual assemblages. These discoveries have prompted researchers to propose that some tyrannosaur species may have been at least occasionally gregarious, traveling together, perhaps hunting cooperatively, perhaps simply tolerating proximity in ways that are not consistent with strictly solitary behavior. The counter-arguments are significant. Crocodilians, some of the most successful large predators in history, are known to aggregate at food sources without being truly social. Modern Komodo dragons will gather at a carcass despite being essentially solitary in their behavior. Shared death events at water sources could reflect drought aggregation rather than pack structure. The cognitive complexity required for coordinated group hunting may not have been present in animals whose brain anatomy, as estimated from endocasts, appears to have been more similar to that of crocodilians than to that of modern birds and mammals. The debate continues. New specimens are found, new analytical methods are applied to old specimens. The picture of tyrannosaur behavior remains fascinating, contested, and unresolved. This is in many ways characteristic of paleontology as a science. The fossil record is magnificent in its scope and fragmentary in its detail. Every specimen is the remnant of a single individual from a population that may have numbered in the thousands or millions. Every discovery is simultaneously an addition to knowledge and a reminder of how much we do not know. Now, let us ask a question that seems almost absurd, but that contains within it a genuine inquiry into the nature of our biology and the conditions that shaped it. Could a human being have survived in any of the prehistoric eras we have described? The question is worth taking seriously because the answer forces us to think carefully about what our bodies actually require. Consider first the Hadean Eon, the first 500 million years of Earth's existence. The surface temperature was far above what human physiology can tolerate. The atmosphere contained no free oxygen. The radiation environment was extreme. The landscape was geologically unstable in ways that would make any modern environment seem placid. A human being transported to the Hadean Earth would survive for seconds, perhaps minutes. The cause of death would be difficult to specify. Oxygen deprivation, thermal injury, radiation damage, and atmospheric toxicity would all be operating simultaneously. The Archean Eon, for all its billions of years of duration, offers little improvement. Atmospheric oxygen remained essentially absent until near the end of the Archean. Surface temperatures, while lower than the Hadean, were still significantly higher than today. The ultraviolet radiation environment remained lethal for complex organisms at the surface. Our bodies depend on oxygen for every cellular process. Without it, unconsciousness follows within minutes and death within a very short time thereafter. No adaptation, no technology, and no evolution could make a human being viable in an oxygen-free atmosphere without artificial life support. The Proterozoic Eon, the era that followed the Archean from about 2 billion to 540 million years ago, saw the gradual rise of atmospheric oxygen. But the process was slow and uneven. The late Proterozoic, in particular, was characterized by a series of extreme glaciation events, the snowball Earth episodes, during which ice may have extended to equatorial latitudes. During these periods, mean global temperatures may have fallen to minus 40 degrees Celsius or colder. Human beings without the benefit of fire, shelter, and clothing of animal origin can survive for only a few hours in temperatures significantly below freezing. The snowball Earth intervals of the Proterozoic would have been catastrophically cold, even if the atmospheric chemistry had been survivable. The early Paleozoic, the Cambrian through the Devonian, presents different challenges. Atmospheric oxygen was rising and by the Devonian had reached levels at least approaching modern values. But the land was being colonized by life that included, as we have discussed, arthropods of extraordinary size. A forest inhabited by centipedes 2 meters long and dragonfly-like insects with wingspans exceeding half a meter would present hazards to an unequipped human that are difficult to assess but difficult to dismiss. Perhaps more importantly, the food available on land in the early Paleozoic would have been limited. Land life was simple and low in nutritional density by modern standards. There were no fruits, no seeds of the kind that grasses and flowering plants produce, no agricultural crops. A human being dropped into the Devonian landscape would be a dietary generalist in an ecosystem that had no evolutionary relationship to Homo sapiens. Survival would require improvisation of the most radical kind. The Carboniferous and Permian, as we have described, had elevated atmospheric oxygen higher than today. This sounds appealing, but the consequences for a human respiratory system are complex. At oxygen concentrations above about 30%, organic materials become dramatically more flammable. The Carboniferous world, during periods of elevated oxygen, would have been prone to wildfires on a scale exceeding anything in the modern world. Breathing hyperoxic air over extended periods carries its own physiological risks. The advantages and disadvantages would depend heavily on local conditions. The Mesozoic, the era of the dinosaurs, is where popular imagination tends to place the human time traveler. and in some respects the Mesozoic world would have been more familiar than earlier periods. By the Cretaceous, flowering plants had diversified extensively. Some of the plants would have been edible. Atmospheric composition was broadly similar to today. Oxygen within a range that human lungs could use, carbon dioxide somewhat higher than present, but not immediately toxic. Temperatures were warmer on average, particularly at high latitudes. But the fauna of the Mesozoic presents obvious challenges. A world in which the apex terrestrial predators were animals capable of reaching lengths of 12 or 13 meters and masses of several metric tons is not a world in which unprotected humans would be safe. It is worth noting, however, that the Mesozoic also contained vast numbers of smaller dinosaurs, crocodilians, lizards, and early mammals, a diverse fauna, not simply a world of giants. The ecosystems were complex. Whether a human being could have found a viable ecological niche within them, avoiding the large predators while exploiting the available food resources, is a genuinely interesting speculative question. What is certain is that the Earth of the Mesozoic was not designed for human beings. Our species emerged in conditions, the fluctuating climate and open savanna environments of the late Cenozoic, that shaped our physiology, our social complexity, and our technological aptitude. We are children of the ice ages as much as of the preceding millions of years of primate evolution. Remove us from those conditions by any significant amount, and survival becomes an increasing challenge. Now let us turn to a different kind of question. One about the upper limits of life itself. How large can a living organism be? The largest animals we know today give us a sense of the range. On land, the African bush elephant reaches masses of up to 13 metric tons, the largest living terrestrial animal. In the ocean, the blue whale can reach lengths exceeding 30 meters and masses, approaching 200 metric tons. The largest animal known to have existed in Earth's history, as far as we can determine. Why the difference between land and water? The answer is physics. In an aquatic environment, buoyancy counteracts much of the downward force of gravity. A body that would collapse under its own weight on land can be supported by water. The structural requirements for a large aquatic body are dramatically less severe than for an equivalent mass on dry land. This is why the largest animals have always been marine. Even the largest Mesozoic dinosaurs, impressive as they were smaller than the blue whale on land. The limits are set by the relationship between body mass and structural strengths. As an animal grows larger, its volume and therefore its mass increases as the cube of its linear dimensions, while the cross-sectional area of its bones and muscles increases only as the square. Doubling the linear dimensions of an animal eightfolds its mass, but only quadruples the structural capacity of its skeleton. This relationship, which Galileo first analyzed in the 17th century, means that there is an absolute upper limit to the size of a land animal before its skeleton fails under its own weight. The great sauropod dinosaurs of the Mesozoic approached this limit. Their anatomy shows extreme adaptations for reducing skeletal mass, hollow bones, pneumatized structures that reduced weight without sacrificing structural integrity. Even so, the largest sauropods were likely operating near the physiological limits of terrestrial animal size. Could life on other planets with different gravity, produce larger organisms? In principle, yes. On a planet with gravity significantly lower than Earth's, the structural constraints would be relaxed. An animal built on the same biochemical plan as an elephant, but living on a world where gravity was half as strong, could theoretically reach masses many times greater. Conversely, on a world with stronger gravity, the maximum size for terrestrial organisms would be smaller. But what about the absolute upper limit? Not for an animal on a planet's surface, but for any biological entity whatsoever. Consider the possibility of a life form that exists not on a planetary surface, but in space itself. If an organism could dispense with gravity as a constraint, drawing energy from starlight, acquiring mass from cosmic dust and gas, reproducing by division, what limits its size? The answer, perhaps surprisingly, is metabolic heat. All biological processes generate heat as a byproduct. The heat generated by the metabolism of a large organism must be dissipated, radiated away, or it will raise the organism's internal temperature to a lethal level. The surface area available for heat dissipation grows more slowly than the volume of the organism, meaning that larger organisms face increasingly severe thermal management challenges. Physicist Max Kleiber identified in the 1930s that metabolic rate scales with body mass to the power of approximately 3/4, a relationship that holds across an enormous range of animal sizes and that reflects, at least in part, the geometry of heat management. At sufficiently large scales, the metabolic rates required to sustain complex biological functions would produce heat that the organism could not radiate away. This sets an upper bound on the size of complex biological systems, at least those based on the same thermodynamic principles as life on Earth. There is another limit, equally fundamental, that applies to any organism large enough that information cannot travel quickly across its body. The nervous systems of animals rely on electrical signals traveling at roughly 100 meters per second. For a blue whale, the lag between stimulus and response is already measurable. For a hypothetical organism the size of a solar system, a nerve impulse would take thousands of years to cross from one end to the other. The organism could not function as an integrated unit in any meaningful sense. Any intelligence it possessed would think at a pace incomprehensibly slow by human standards. Some thinkers have proposed that the universe itself might be considered a kind of living system, one that processes information, maintains structured complexity, and evolves over time through the formation and death of galaxies and stars. This is a philosophical rather than a scientific claim, but it has the virtue of forcing us to question what we mean by life and what properties we consider essential to it. If life is defined primarily by complexity, information processing, and self-replication, then the boundaries of the concept may be far broader than the familiar forms we observe on Earth. What we can say with confidence is that the known universe is vastly larger than any biological organism, and that the physical laws governing both are the same. The materials from which life builds itself were forged in stellar interiors and distributed across the galaxy by stellar death. We are, in the most literal sense, products of cosmic processes, beings assembled from stardust, animated by chemistry, shaped by the particular history of this particular planet. Which brings us to a question that may be the most fundamental of all. Why does life use the chemistry it does? If you look at the composition of any living thing on Earth, a bacterium, a blue whale, a flowering plant, you find that the vast majority of the dry mass is made of just 6 elements: carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. These 6 elements, sometimes abbreviated as CHNOPS, account for roughly 98% of the mass of all living organisms on Earth. Every protein, every nucleic acid, every lipid membrane, every sugar is built primarily from these 6 elements. Why these 6? Is this an arbitrary choice of evolution, a contingency of the particular history of life on Earth? Or is there something necessary about this selection? One approach is to compare the 6 elements to the cosmic abundance of elements, the proportions in which different elements occur throughout the universe. Hydrogen is the most abundant element in the cosmos. Carbon ranks 4th by mass in the universe. Oxygen ranks 3rd. Nitrogen ranks 7th. So 3 of our 6 life-building elements are among the cosmically most abundant. But the pattern breaks down. Helium is the second most abundant element in the universe, but it plays no role in biochemistry. It is chemically inert, essentially incapable of forming stable chemical bonds with other elements under ordinary conditions. Neon and argon, also abundant, share this characteristic. The noble gases, however prevalent in the universe, are chemically useless to life. Phosphorus is far less cosmically abundant than the life-building elements we might expect. It ranks perhaps 18th or 20th in terms of cosmic abundance, and yet phosphorus is indispensable to life as we know it. It forms the backbone of DNA and RNA, the molecules that carry genetic information and direct the synthesis of proteins. It is a component of adenosine triphosphate ATP, the primary energy currency of the cell, the molecule that stores energy in a chemically accessible form and releases it to power cellular work. Without phosphorus, the information storage and energy transfer systems of life as we know it would fail. Why phosphorus? The specific chemical properties of phosphate groups, the phosphorus-oxygen structures that appear throughout biochemistry, make them uniquely well suited for certain biological functions. The stability of phosphate esters, combined with their reactivity under enzymatic conditions, makes them ideal for both storing information in nucleic acids and transferring energy in ATP. No other element plays these roles as well in the chemistry of water-based life. But this raises a profound question. If the chemistry of life on Earth was shaped partly by contingency, by which molecules were available in the right concentrations at the right times in the right places, when life was getting started, then life on other planets might have developed around different chemical foundations. If the early ocean where life originated on some other world happened to have different composition, different temperature, different pH, then the molecules that became the basis of life there might be completely unlike those of terrestrial biology. This is the domain of astrobiology, and within it, a fascinating subfield. The study of alternative biochemistries, the most obvious alternative to consider is the question of solvent. Life on Earth uses water as its universal solvent, the medium within which chemical reactions occur, molecules are transported, and cellular structures are maintained. Water has properties that make it extraordinarily well suited for this role. It is liquid over a wide range of temperatures under Earth's conditions. It is a polar molecule, which means it is an excellent solvent for the ionic and polar compounds that biochemistry relies on. Ice floats, a consequence of water's unusual hydrogen bonding, which means that freezing bodies of water form an insulating layer at the surface rather than freezing solid from the bottom up. But water is liquid only between 0 and 100 degrees Celsius at standard pressure. Throughout the universe, the vast majority of planetary surfaces are either far colder or far hotter than this range. On most planets, water would be either ice or steam. Ammonia has been proposed as an alternative biological solvent since at least the 1950s, when biologist J.B.S. Haldane discussed the possibility ammonia is liquid at temperatures between -78 and -33 degrees Celsius at standard pressure, a range appropriate for the conditions found on some cold outer solar system moons. Like water, ammonia is a polar molecule and a good solvent for many biologically relevant compounds. Many chemical reactions that occur in aqueous solution have ammonia-based analogues. It has even been proposed that amino acid-like and nucleic acid-like molecules could form and function in liquid ammonia, though the chemistry would be very different in detail. A life form based on liquid ammonia would inhabit an environment catastrophically inhospitable to any Earth organism. Water for such a life form would be a corrosive acid, its ability to disrupt hydrogen bonds in ammonia-based structures, making it as destructive as strong acid would be to us. The temperatures required for liquid ammonia to serve as a solvent are also far below human tolerance. Any encounter between an ammonia-based organism and the conditions of Earth's surface would be immediately lethal to the organism. Other proposed alternative solvents include liquid methane, as found in the lakes and seas on Saturn's moon Titan, and hydrogen fluoride. Each has interesting chemical properties that, under the right conditions, might support some form of complex chemistry. Titan is particularly intriguing because it actually has stable liquid on its surface, methane, ethane, lakes, and seas, and because it has a complex organic chemistry driven by ultraviolet photolysis of atmospheric methane. Whether that chemistry can support anything that deserves the name life is one of the most compelling unanswered questions in astrobiology. The question of alternative carbon chemistry, whether carbon is the necessary basis for all life, or whether other elements could substitute centers primarily on silicon. Silicon occupies the same column of the periodic table as carbon, which means it has the same valence. It can form 4 chemical bonds simultaneously like carbon. Silicon can form chains, rings, and complex 3-dimensional structures. In the 19th century, the astrophysicist Julius Scheiner was among the first to suggest that silicon might serve as the basis for life on worlds where carbon-based chemistry was not viable. Silicon has real advantages for certain environments. Its compounds are thermally stable at temperatures that would destroy organic molecules. Silicon-oxygen bonds are among the strongest in chemistry, making silicate structures extremely durable. If life must function at temperatures of several hundred degrees Celsius, silicon chemistry might have advantages that carbon chemistry lacks. But silicon also has profound limitations. The silicon analog of carbon dioxide, silicon dioxide is not a gas but a solid under ordinary conditions. It is quartz, one of the most common minerals in the Earth's crust. A silicon-based organism that oxidized its energy sources, the way we oxidize glucose, would produce silicon dioxide as a waste product, breathing out sand in effect. The respiratory and excretory systems of such a life form would have to be radically different from anything in Earth biology. Silicon also forms polymers less readily than carbon, and the resulting polymers are generally less complex. Carbon's ability to form long, stable chains with diverse functional groups hanging off them, the chemical property that makes possible the extraordinary diversity of organic molecules, has no precise silicon analog. Silicon chemistry can produce complex structures, but probably not structures of the same diversity and functional versatility as carbon biochemistry. This may be a significant constraint on the complexity of any life based primarily on silicon, and yet the combination of silicon and oxygen in extreme high-temperature environments without free oxygen in the atmosphere might produce a very different kind of chemistry from what we know. Silicate minerals at very high temperatures deep in a planetary interior might support chemical reactions of a kind that we have barely begun to imagine. Some researchers have speculated about life forms that function at the temperatures and pressures of the lower mantle, 1,000 kilometers beneath the surface, using chemistry radically unlike anything on the surface. The idea is difficult to test and easy to dismiss. But the history of biology is full of environments that were declared incompatible with life until life was found there. Hydrothermal vents on the deep ocean floor, the interior of Antarctic ice sheets, the clouds of hot acidic vapor above Yellowstone's geysers, the bedrock 2 or 3 kilometers beneath Earth's surface where bacteria live on hydrogen produced by geochemical reactions in the rock. Life on Earth has demonstrated a persistent ability to colonize environments that prior thinking would have ruled out. This should make us humble when we attempt to define the outer limits of what is chemically possible for life elsewhere. The truly exotic possibilities life forms based on boron hydrates, on hydrogen fluoride solvents, on entirely different structural chemistries, strain our chemical intuition but do not necessarily violate physical laws. Boron hydrates can form complex cage-like structures with interesting chemical properties. Under the right conditions, Low temperature, reducing atmosphere, absence of the reactive chemicals that would destroy them in an Earth-like environment. Such structures might support chemistry of considerable complexity. On Earth, these compounds are explosively reactive in contact with oxygen or water. On a world with a different atmospheric composition, they might be perfectly stable. The universe contains approximately 2 trillion galaxies, each containing hundreds of billions of stars, most of which appear to have planetary systems. The number of planetary bodies in the observable universe is a number that the human mind cannot genuinely grasp. Some estimates put it in the range of a septillion, which is 1 followed by 24 zeros. Within that vast catalogue of worlds, conditions vary across a range that dwarfs anything we find in the solar system. Temperatures from near absolute zero to thousands of degrees. Gravity from fractions of Earth's surface gravity to dozens of times greater. Atmospheric compositions from pure hydrogen to concentrated sulfuric acid. If life can arise wherever the right chemical conditions exist, if the emergence of self-replicating, evolving chemical systems is something that chemistry does naturally when given enough time and the right ingredients, then life in the universe may be nearly as diverse as the range of environments it inhabits. Some of that life would be immediately recognizable as life carbon-based water solvent operating on biochemical principles broadly similar to our own, different in the specific molecules, different in its evolutionary history, but fundamentally familiar in its logic. Some of it would be profoundly alien, operating at temperatures where our biology would instantly denature, using solvents that would dissolve our cells, built on polymer chemistry that no terrestrial biologist has ever worked with in a living context, reproducing by mechanisms we might not immediately recognize as reproduction. And some of it might be so different from everything in our conception of life that we might not recognize it as life at all. We might walk past it, or fly past it, or observe it from orbit, without understanding that what we were seeing was an organism. The history of science is, in many respects, a history of expanding the boundaries of what we thought possible. The discovery that life exists in the deep ocean around hydrothermal vents, ecosystems driven not by sunlight, but by chemical energy, thriving in pitch darkness and extreme pressure, extended the known boundaries of life on Earth. The discovery of microorganisms living in the bedrock kilometers beneath the surface at temperatures and pressures that were long thought incompatible with biology extended them further. Every generation has found that life is more tenacious, more chemically inventive, and more willing to inhabit extreme environments than the previous generation believed. This pattern gives reason for optimism about the broader question of life in the universe, and it gives reason for humility about our ability to predict where and in what form life will be found. All of the chemistry of life, the carbon and hydrogen and oxygen and nitrogen, the phosphorus and sulfur, the water and the organic molecules, was assembled over billions of years from material scattered across the galaxy by dying stars. The atoms in our bodies were synthesized in stellar interiors and distributed through space by supernova explosions. Some of the hydrogen in the water in our cells is as old as the universe itself, formed in the first minutes after the Big Bang. We are, in the most literal sense imaginable, made of ancient things. The story of prehistoric Earth is not simply a catalogue of extinct organisms and vanished landscapes. It is the story of a planet learning to sustain life, of chemical systems growing more complex, of ecological relationships deepening, of catastrophe and recovery cycling over and over, through geological time. Every mass extinction in Earth's history was followed by a recovery, often more diverse than what had preceded it. The Permian extinction, devastating as it was, gave rise eventually to the extraordinary diversity of the Mesozoic. The end Cretaceous extinction, which removed the non-avian dinosaurs, opened ecological space into which mammals diversified, ultimately producing the lineage that gave rise to us. we tend to think of extinction as failure. But in the long perspective of planetary history, extinction is also opportunity. It clears space, it removes ecological constraints, it allows lineages that had been suppressed or marginalized to expand and explore new possibilities. The Earth we live on now is the product of 4.5 billion years of this process. Every organism alive today is the product of an unbroken chain of ancestors extending back to the earliest life on this planet, to those simple microbial mats that carpeted the floors of Archean oceans 3.5 billion years ago. That chain has never been broken. Through every catastrophe, through every extinction event, through every glaciation and volcanic episode and asteroid impact, the thread of life has persisted. We stand at a particular moment in that history. Our species, Homo sapiens, has existed for perhaps 300,000 years, an eye blink on the geological timescale. In that brief span, we have shaped the planet in ways that will be visible in the geological record for millions of years to come. The lesson of deep time is not that the Earth is fragile. It is not the Earth as a geological entity will persist long after any mass extinction, long after any climate perturbation, long after any human intervention. Life, in some form, will almost certainly persist with it. The question that deep time poses to us is not about the Earth's survival. It is about the kind of world we choose to inhabit and to live. Every era in Earth's history has been shaped by the chemistry and physics and biology of its time. Ours is the first era shaped significantly by the choices of a single species. What we make of that responsibility is the question our era will be remembered for, assuming there is anyone or anything to remember it. The chronicle of prehistoric Earth does not end with the emergence of Homo sapiens. It continues. We are not the conclusion of the story, but a chapter within it. An extraordinary, self-aware chapter, capable for the first time of reading the record of what came before and of making choices that will shape what comes after. Whether we prove equal to that responsibility remains, at this moment, an open question. Consider, for a moment, the particular problem of measuring time. Human beings are calibrated to think in years, decades, generations. A century feels long. A millennium is the edge of historical imagination. 10,000 years, the span of recorded civilization, feels ancient, vast, almost incomprehensible. And yet 10,000 years is nothing against the backdrop of Earth's history. If the entire 4.5 billion year history of the planet were compressed into a single calendar year, all of recorded human history would occur in the last 14 seconds of December 31st. This compression is not merely a rhetorical device. It reflects something real about the nature of geological time, about the extraordinary patience of the processes that shape planets and give rise to life. The Grand Canyon, one of the most vivid geological spectacles on Earth, exposes 2 billion years of rock in its walls. Standing at the rim and looking down is a form of time travel, each visible stratum representing millions of years of deposition, erosion, and geological activity. The Colorado River cut that canyon over the last 5 to 6 million years, and that timescale itself is longer than the entire existence of our genus. Geological thinking requires a fundamental reorientation of perspective. The question is not why things change so slowly, but rather why we expect the pace of geological change to match the pace of human experience. They operate on entirely different timescales, governed by entirely different forces. With that perspective in mind, let us return to the early solar system and to a process that continued long after the planets had formed. The period known as the Late Heavy Bombardment, thought to have occurred roughly 4 to 3.8 billion years ago, was a time when the inner solar system experienced an elevated rate of large impactors. The evidence for this comes from the lunar surface. Many of the Moon's largest impact basins, the dark regions visible to the naked eye from Earth, called Maria, were formed during this interval. Similar bombardment would have struck Earth, Venus, Mercury, and Mars during the same period, though Earth's active geology has erased virtually all direct surface evidence of these ancient impacts. The cause of the late heavy bombardment is still debated. One influential hypothesis, part of a larger model of early solar system dynamics known as the Grand Tack hypothesis, proposes that the giant outer planets underwent significant orbital migrations in the early solar system. As Jupiter and Saturn moved, their gravitational influence destabilized vast numbers of smaller bodies in the outer solar system, sending them on trajectories that brought them into the inner solar system as a wave of impactors. If this model is correct, the conditions of the early inner solar system were shaped as much by the dynamics of the giant planets as by the local processes at each individual planet. For Earth, the late heavy bombardment would have had two apparently contradictory effects. On one hand, large impacts can vaporize surface water and temporarily sterilize the upper crust. Early life, if it had begun, would have been periodically threatened by impacts that would have heated the surface to temperatures lethal to all but the most heat-tolerant organisms. On the other hand, the comets and water-rich asteroids that made up a portion of the impactors may have delivered significant quantities of water and organic material to the early Earth. Some portion of the water in Earth's oceans, and perhaps some of the organic precursors to life, may have been delivered from space during this period. The origin of Earth's water is itself a fascinating and unresolved question. The inner solar system close to the young sun, where temperatures were high, was largely depleted in water during the earliest stages of planet formation. The regions where water ice could have condensed onto solid grains, the so-called frost line, lay beyond the asteroid belt. Yet Earth has abundant water. Where did it come from? The answer almost certainly involves some combination of water-bearing asteroids from the outer asteroid belt, comets from the outer solar system, and perhaps water locked into the minerals of the accreting material from the start. The isotopic signature of Earth's water. The ratio of heavy hydrogen to ordinary hydrogen points most strongly to an asteroidal source rather than cometary, though the details remain active areas of research. Now, having established the early physical history of Earth, let us think more carefully about the transition from chemistry to biology, from complex molecules to living systems. This transition remains one of the deepest unsolved problems in science. We know that life on Earth is based on two classes of polymer: nucleic acids, which store and transmit genetic information, and proteins, which carry out the structural and catalytic functions of the cell. The relationship between these two classes is deeply circular. Proteins are required to copy nucleic acids, but the information needed to make those proteins is stored in the nucleic acids. Which came first? The RNA world hypothesis proposes a resolution to this chicken and egg problem. RNA, ribonucleic acid, can both store genetic information, like DNA, and catalyze chemical reactions, like proteins. In this model, the earliest life was based on RNA, which served both functions simultaneously. DNA and proteins were later additions that took over the information storage and catalytic roles, respectively, because they performed those functions more efficiently. The discovery of ribozymes, RNA molecules with catalytic activity in the 1980s by Thomas Cech and Sidney Altman, for which they shared the Nobel Prize in Chemistry in 1989, provided strong experimental support for this idea. But even if RNA was central to early life, the question remains, how did RNA first form? RNA is a complex molecule. Its component parts, the sugar ribose, the nitrogenous bases, and the phosphate groups must be assembled in a precise way to create a functional polymer. Synthesizing RNA from simpler precursors in a test tube requires carefully controlled conditions that seem difficult to achieve in a natural environment. Much recent research has focused on finding plausible geochemical settings where the required chemistry might spontaneously occur. Warm ponds subject to cycles of wetting and drying, hydrothermal vents, ice surfaces where molecules could be concentrated and protected. The discovery of complex organic molecules in meteorites, amino acids, nucleobases, sugars has suggested that the raw materials of life may be widespread in the universe and may have been delivered to the early Earth in abundance. Whether these delivered molecules played a direct role in the origin of life, or whether life assembled its building blocks entirely from terrestrial chemistry, remains unclear. Possibly both pathways contributed. What we can say is that life arose relatively quickly in geological terms, after the Earth's surface cooled enough to allow liquid water to persist. The oldest known microfossils and chemical signatures of life date to roughly 3.7 to 3.8 billion years ago, within a few 100 million years of the end of the late heavy bombardment. This rapid emergence, combined with the persistence of life through subsequent catastrophes, suggests that once the conditions for life are established, the transition from chemistry to biology may not be as improbable as it sometimes seems. Or perhaps we are the exception, the singular unlikely case in an otherwise lifeless universe. And the rapid origin of life on Earth simply reflects the fact that if it had not originated quickly, we would not be here to observe it. This selection effect argument, sometimes called the anthropic principle in one of its forms, is logically valid but philosophically unsatisfying, because it cannot be distinguished empirically from a universe in which life originates easily and often. The only way to resolve this question is to find life elsewhere. If life has arisen independently on other worlds within our own solar system, on Mars, on Europa, on Enceladus, then the emergence of life is probably a common cosmic phenomenon. If, after exhaustive search, life is found only on Earth, the question of its rarity becomes more urgent. Mars is a particularly compelling case. Evidence from orbital observations, landers and rovers has established that Mars had liquid water on its surface for at least hundreds of millions of years in its early history, long enough for life to potentially have arisen. The Martian surface today is cold, dry, and irradiated, but the Martian subsurface may contain liquid water in pockets and aquifers. Methane has been detected in the Martian atmosphere at trace levels. Methane that must be produced by either geological or biological processes, since ultraviolet radiation destroys it quickly and it must be continuously replenished. The source of Martian methane is one of the most intriguing unsolved questions in planetary science. Europa, the ice-covered moon of Jupiter, has a global subsurface ocean of liquid water, kept from freezing by tidal heating from Jupiter's gravity. The ocean floor of Europa may have conditions, pressure, temperature, chemical gradients broadly similar to the deep-sea hydrothermal environments on Earth, where dense communities of organisms thrive in the complete absence of sunlight. Enceladus, a moon of Saturn, actively vents water vapor and organic material into space through geysers near its south pole, direct evidence of a subsurface ocean and an opportunity to sample that ocean without landing on the moon's surface. These worlds are not hosts to complex, multicellular life in any scenario that current science can envision. The evolutionary history required to produce cells with nuclei, then multicellular organisms, then organisms capable of complex behavior, took billions of years on Earth and required a specific combination of conditions that may not be present elsewhere in the solar system. But microbial life organisms, no more complex than the bacteria that dominated Earth for most of its history, might exist or have existed in these environments. If it does, the implications would be profound. They would tell us that life is not a singular accident confined to a single planet, but a recurring phenomenon, chemistry's answer to the problem of extracting energy from environmental gradients and maintaining organized complexity against the tendency of systems to disorder. It would mean that the emergence of life is not a miracle, but a natural consequence of the universe's chemistry operating over long enough timescales in appropriate environments. The question of how common life is shapes our interpretation of everything else we know about the universe. If life is rare, Earth's history is a unique and precious anomaly. If life is common, Earth's history is one example of a process that has played out billions of times across the galaxy. We do not yet know the answer, but we are, for the first time in history, developing the tools to begin answering it. Space telescopes capable of detecting the chemical signatures of planetary atmospheres from light years away, spacecraft designed to search for life in the liquid water environments of icy moons, robotic explorers drilling into Martian rock in search of biosignatures, ground-based telescopes scanning the radio spectrum for signals that might carry the imprint of intelligence. The next few decades may bring answers to questions that have occupied human thought for centuries, or they may bring only more refined uncertainty. But the search itself, the application of scientific method and technological capacity to the oldest of human questions, represents one of the most significant intellectual endeavors of our species. We began this chronicle with an ending, the catastrophe that brought the age of the dinosaurs to a close 66 million years ago. We end it with the beginning of our capacity to ask rigorously and systematically whether the story of life on Earth is the universe's story as well, or merely our own. The Earth, 4.5 billion years old, has hosted life for most of its history. That life began as something barely distinguishable from chemistry and became, over time, capable of contemplating its own origins, capable of reading the record preserved in rock and ice and starlight, capable of wondering whether the same story has unfolded elsewhere, in other solar systems, around other stars, in other galaxies beyond the reach of our current telescopes. Whatever the answer turns out to be, the question itself is remarkable. It took 4.5 billion years for a planet to produce an organism capable of asking it. We are that organism. And the inquiry continues.